Hi there, welcome to this video. So we're going to look at respiration stage 2, which is the link reaction. It follows on from the respiration stage 1 video on glycolysis, so you might want to watch that first. So at the end of glycolysis, we've made a molecule called pyruvate. Well, we've made two pyruvate molecules per glucose. Okay, so we've got two pyruvate molecules from the previous stage. Now, the previous stage, glycolysis, actually happened in the cell's cytoplasm. Okay, now the next stage, if this is our cell, this is our nucleus, the next stage is going to happen in the mitochondria. So these pyruvate molecules need to move into the mitochondria. Now you might remember that the mitochondria has a smooth outer membrane and a folded inner membrane. Now for this stage, the part of the mitochondria that we're interested in is the part in the middle. In the middle of the mitochondria is called the matrix. So that space in the middle of the mitochondria is where link reaction is going to happen. So the two molecules of pyruvate from glycolysis from stage 1 are going to make their way into the mitochondria, into this middle section here called the matrix, ready for the link reaction and the Krebs cycle, which is stage 3. Okay, so we're going to go on to link reaction. So our starting point for the link reaction is pyruvate. Now remember that all of this is times 2 because we made two pyruvate molecules per molecule of glucose that we split in glycolysis. Okay, so in the link reaction, we're starting off with pyruvate. Think about how many carbons pyruvate had. So glucose had 6, and glucose got split into 2 3s. So pyruvate has 3 carbons. Now, the first thing that happens is we're going to use another one of these NAD molecules. So NAD, if we remember, let's do a recap. NAD is a coenzyme. You can also call it an electron carrier. And its job in respiration is that it carries hydrogen atoms to stage 4. Okay, so it picks them up in the earlier stages and takes them to stage 4. So NAD is going to have a reaction with pyruvate and pick up some hydrogen to become reduced NAD. Instead of saying reduced NAD, you could call it NADH. That's the same molecule. Now, there is actually another way you can write them, which I'll show you just in case it comes up. NAD is sometimes called NAD+, and if you were using that format, then you would say that it makes NADH plus H plus. Okay, so it's just another way of writing it. If I were you, I would just say NAD to reduce NAD, but I just want you to have seen that version in case they use it in the exam. It's nice to have obviously seen it. Okay, so what's happening there on the tiny scale is that the pyruvate is giving hydrogen to the NAD, producing reduced NAD. Don't forget the oil rig. The reason we call it reduced NAD is because reduction is gain. Okay, so the NAD has gained hydrogen, hence it's called reduced NAD. The pyruvate at the same time, the pyruvate has been oxidized because the oil part stands for oxidation is loss. So the pyruvate has given the hydrogen to NAD. So the pyruvate has been oxidized. Okay, and the NAD has been reduced. Okay, the next thing that happens here is that we lose a molecule of carbon dioxide. So it produces a molecule of carbon dioxide. Now, carbon dioxide is CO2, so it obviously has one carbon. So this 3-carbon molecule here, pyruvate, if we make a molecule of carbon dioxide and it gets released, how many carbons are now left? Just 2 carbons. So we've now produced a new molecule. Our new molecule here is called acetate, and our acetate now only has 2 carbons. Okay, so we started with pyruvate, which had 3 carbons. The pyruvate gave a hydrogen to NAD to reduce NAD. We released a molecule of carbon dioxide, and we're left with a molecule called acetate, which has 2 carbons. The next stage that happens is that another molecule actually gets added in. So we draw that like this. So the enzyme that's going to be added in is called coenzyme A. Sometimes you might see it written just CoA. Okay, and that produces, if I just go out of the board and back in, I think it just shrinks it a little bit. Just thinking I'll have more room to the right. That's all. Let's move that up. So acetate plus coenzyme A together make acetyl coenzyme A. Okay, this didn't contain any carbons, so the 2 carbons that we have there will remain. It's still going to be a 2-carbon molecule. Okay, and that's going to form our starting point ready for the Krebs cycle. Remember that all of this happens twice. Okay, a little bit more language about how we can describe it. When we produce the molecule of carbon dioxide just here, you can say here that the pyruvate is decarboxylated. Decarboxylated means that carbon is removed, okay? So it's lost a carbon. Here, because the pyruvate lost a hydrogen to give to the NAD, we could say that the pyruvate, as well as being oxidized, we could say that it's dehydrogenated. Dehydrogenated. Okay, so as well as saying that the pyruvate is oxidized, we can also say that pyruvate has been dehydrogenated. So it's oxidized because it lost a hydrogen, and because it lost a hydrogen specifically, we can call it dehydrogenated. So there's just a few extra words there that you can use. Each of these stages would be catalyzed by enzymes, but there's no requirement to learn the names of the enzymes. Definitely don't try and do that. It'll be too much to learn. Okay, once the carbon dioxide has been released here, you know the original respiration equation that you did at GCSE. On the right-hand side, you'll have put carbon dioxide in water. So that's the carbon dioxide. That's a waste product. It simply gets breathed out. And we'll look at where we form the water in stage 4. Okay. When we talk about the acetate there, you might have learned that that was called acetyl. It's because this is just a simplified version, okay, and there's actually lots of stages. So as long as you just know that there's an intermediate there, don't worry about which of the names that you use. It's not a problem. So for each pyruvate that we have, we make 2 NADs. So here, sorry, I'm going to add that in then. Where I've put NAD there, there's 2 NADs, and then they get made into 2 reduced NADs. When we said it was giving hydrogen, we could say it's giving 2 hydrogen, okay, just to make sure you've got your numbers right. This section doesn't make any ATP molecules. Okay, so there's no ATPs to add up here, but it is important that we've got 2 acetyl coenzymes enzyme A's, which are the starting point of the Krebs cycle, and we've got some reduced NAD, which are really important for stage 4. So let's look at a summary of this. So the link reaction starts off with the pyruvate, okay, and the end point is acetyl coenzyme A. You can call it just CoA for short. There's no ATP produced. There's actually 2 pyruvates at the start and therefore 2 acetyl coenzymes A's at the end. Each pyruvate makes 2 reduced NAD, so total it makes 4 reduced NAD molecules. You remember these reduced NAD molecules are going to be taken to stage 4, okay, ready to make more ATP. So the reduced NADs go off to stage 4. The acetyls are going to go off to the Krebs cycle, and we've not, and we've not got any ATPs at this stage for counting. And that's the end of this video.